Let's start with section 1. Most cell division results in genetically identical daughter cells. Before we get to know what cell division is, what would be the key roles of cell division? The first role of cell division is asexual reproduction. This is applied to prokaryotes and unicellular eukaryotes since cellular division process gives rise to a new organism or another cell. The second role is growth and development. The process helps organisms to develop from a single cell into the fertilized egg. The last role of cell division is tissue renewal by replacing the dead cells into new cells. Now, let's look at some vocabulary that we should know before we learn more about cell division. Genome is a cell's DNA, or the genetic information. Chromosome is the packaged form of DNA molecules. Chromatin is the entire complex of DNA and proteins that is the building material of chromosomes. Somatic cells are all of the body cells except for the reproductive cells. Lastly, gametes are reproductive cells. So, what are genomes exactly? Genome is the sum of all genetic material of an individual. Prokaryotic genome is often a single DNA molecule, and eukaryotic genomes are usually made of multiple DNA molecules. DNA must be copied and separated before the cell divides so that each daughter cell can have a complete genome. Chromosome can be known as a long DNA molecule. Eukaryotic chromosomes consist of one very long linear DNA molecule associated with many proteins. The DNA molecule carries hundreds to thousands of genes. Chromatin is made of a complex of DNA and proteins. Its degree of condensation varies during the process of cell division. Somatic cells are all kinds of cells except for reproductive cells. Each cell contains 46 chromosomes made of two sets of 23, one set inherited from each parent. On the other hand, gametes are reproductive cells, which includes sperms and eggs. Human gametes have one set of 23 chromosomes. Until now, we've been learning about the most basic concept for the background of cell division. Let's get a little bit deeper, shall we? These are other vocabularies. Sister chromatids are joint copies of the original chromosome. Centromere is where a chromatid is attached to its sister chromatids. Mitosis is the division of the genetic material in the nucleus. Lastly, cytokinesis is the division of the cytoplasm. Sister chromatids join a centromere, which is a region made up of repetitive sequences in the chromosomal DNA. The attachment is mediated by proteins and gives rise to a narrow waist. And the either side of the centromere is called the arm of the chromatid. This is a preview of cell division. From a fertilized egg, mitosis and cytokinesis produce trillions of somatic cells. In contrast, meiosis produces gametes by reducing two sets of chromosomes into one set. Meiosis in humans occurs in ovaries and testes. As fertilization fuses one set of chromosomes into two, mitosis occurs and the process cycles. From now on, you will learn about the steps of the cell cycle in earnest.
In section 2, the mitotic phase alternates with interphase in the cell cycle. In the first part of vocabulary, there are 8 words. Interphase is phases of cell growth and duplication of chromosomes to prepare cell division. Centrosome is an organelle that organizes microtubules, so it is also called a microtubule organizing center. Ester is radial array of short microtubules around centrosomes. Mitotic spindle is structure made of microtubules, which makes chromosomes move or control them. Kinetochore is a protein complex attached to centromere. It is made in prometaphase first. If microtubules are attached to kinetochore, they are called kinetochore microtubules. However, if they are not, they are called non-kinetochore microtubules. The last is the metaphase plate. It is a plane that is equidistant between the spindle's two poles. Cell cycle has two major phases, interphase and mitotic phase. Interphase occurs first. It includes G1, S, and G2 phases. G stands for gap. G1 and G2 got named gap because when they were first discovered, they seemed inactive. However, we now know that metabolic activities and growth occurred intensely. Then, S stands for synthesis of chromosomes. All interphase steps produce protein and cytoplasmic organelles. However, duplication of chromosomes only occurs in S phase. Mitotic phase occurs next. It can be divided into two parts, mitosis and cytokinesis. Mitosis, which is division of chromosomes, can be divided into five steps: prophase, prometaphase, metaphase, anaphase, and telophase. Before we look at the mitotic phases, there is the second gap. In the G2 phase, the nuclear envelope encloses the nucleus. The nucleus contains one or more nucleoli. Each centrosome contains two centrioles. Duplicated chromosomes in S phase are not condensed yet. In prophase, chromatin fibers become tight and become chromosomes which are observable in light microscope. The nucleoli in the nucleus disappear and duplicated sister chromosomes stick to each other. The mitotic spindle begins to form. It contains centrosomes and its microtubules. Shorter microtubules around centrosomes are called esters. Next is prometaphase. In prometaphase, nuclear envelope fragments and chromosomes become more condensed. On each centromere, kinetochore are formed. Some microtubules are attached to kinetochore and some are not. Microtubules which are not attached to kinetochore lengthen the cell with the opposite one. Then metaphase occurs. Centrosomes are now located in the opposite pole of the cell. All chromosomes make a line on the metaphase plate. All kinetochores are attached to microtubules. Next phase is anaphase. Cohesion proteins between sister chromatids are cleaved, so they are separated and dragged to each pole. The microtubule is shortened by depolymerization. The last phase of mitosis is telophase. As the nuclear envelope and nucleoli reappear, two daughter nuclei form in the cell. Chromosomes become less condensed. Rest of the spindle microtubules are depolymerized. By late telophase, cytokinesis is also underway. Cytokinesis is the division of the cytoplasm, which is the last step of cell division. There are four vocabulary words. Cleavage is process of forming cleavage furrow, and it is a line of cell division that pinch the cell in two. Cell plate is young cell wall instead of cleavage furrow that formed during cytokinesis in plant cell. Origin of replication is a site of replication of chromosomes in binary fission. Mitosis depends on the mitotic spindle a lot. The spindle microtubules elongate by incorporating more subunits of the protein tubulin, which is polymerization, and shorten by losing some unit, which is depolymerization. Enzyme called separase makes cohesion between sister chromatids. The enzyme breaks down in anaphase, which allowing chromosomes to divide in two and separate it. Two mechanisms are in play for this polyward movement of chromosomes. Both of them are involved with motor proteins. The first mechanism is that chromosomes walk along the microtubules, which means after the motor protein passes, chromosomes depolymerize microtubules at their kinetochore. An experiment suggested that chromosomes walk, in other words, microtubules depolymerize from the kinetochore, not from the pole. First, people who made the experiment labeled the microtubules of Purkinje cell in early anaphase with yellow dye. Second, they mark the particular region by eliminating the dye by laser. Third, they compare the lengths of microtubules on the pole side and the kinetochore side. Conclusion of the experiment is that during anaphase in this cell type, chromosomes walk to the pole by depolymerizing microtubules from the kinetochore region, releasing tubulin subunits. Another mechanism is that chromosomes are reeled in by motor proteins at the spindle poles and that the microtubules depolymerize after they pass by these motor proteins at the poles. General consensus is that the ways vary among cell types. In animal cells, cytokinesis occurs by cleavage. Process of cleavage form a cleavage furrow which is made of actin microfilaments and protein myosin. It makes a contractile ring of microfilaments and pinch the cell until the cell divides. In plant cells, as there is an additional layer, cell wall, cell plate formed. Deposition of some cell plate materials become new cell walls for two daughter cells. Next is the type of cell division that prokaryotes use. Binary fission, which means division in half, is the simplest form of cell division. Chromosomes replicated itself on the origin of replication, and the cell just divides.
Alright, now it's section 3. The eukaryotic cell cycle is regulated by a molecular control system. Cell division is important in many parts of animals and plants because it is related to normal growth, development, and maintenance in plants and animals. The frequency of cell division depends on the type of the cell. In the human body, human skin cells divide frequently throughout the lifetime. However, liver cells maintain the ability to divide but do not divide steadily. Instead, they divide during the specific needs, such as repairing a wound. Moreover, some cells do not divide at all when the body gets mature. Muscle cells and nerve cells are those types. Then why are the cell cycles so different? These cell cycle differences are caused by the regulation at the molecular level. Thus, the cell cycles are different. Also, the mechanism of a cell cycle is a big interest, not just to understand the life cycle of the cells, but also to learn how cancer cells manage to escape the usual controls. In the early 1970s, scientists hypothesized that the cell cycle is driven by specific signaling molecules present in cytoplasm. One of the strongest pieces of evidence was the mammalian cell experiment. In the experiment, two mammalian cells in S phase and M phase were fused with a cell in the G1 phase. When a cell in S phase fused with G1, the G1 nucleus entered the S phase, and then the DNA was synthesized. Also, when a cell in M phase fused with G1, mitosis occurred. The experiment allowed us to know that molecules present in cytoplasm during the S or M phase controlled the progression to those phases. Therefore, scientists declared that the cell cycle is directed by the cell cycle control system. Cell cycle control system is a cyclic operating set of molecules in the cell that both triggers and coordinates key events in the cell cycle. The cell cycle is often compared to the washing machine because both of them have proceeded by themselves with built-in clock. However, washing machine's cycle is subject to both internal control and external adjustment, whereas the cell cycle is regulated by certain checkpoints by internal and external signals. Checkpoint is a control point where stop and go ahead signals can regulate the cycle. Major checkpoints are G1, G2, and M phases. During the cell cycle, the fluctuations in the abundance of cell cycle control molecules causes subsequent events in the cell cycle. There are two types of molecules. One is protein kinases, another is cyclin. Many kinases in the growing cells are inactive. To be active, it must be attached to a cyclin. Cyclin is a protein that gets its name from its cyclically fluctuating concentration in the cell. Therefore, this type of kinases are called cyclin-dependent kinases, or CDK. The activity of a CDK rises and falls with changes in the concentration of its cyclin partner. For example, when the concentration of the cyclin is at the peak, the activity of MPF is also at the highest point. MPF is cyclin-CDK complex in frog eggs. You can check that as the concentration of cyclin gets higher, the higher MPF activity. Now we will look more about checkpoints. At the checkpoint, many signals are registered inside and outside the cell. As I mentioned earlier, three important checkpoints are G1, G2, and M phases. Mostly, the G1 checkpoint is the most important step. If a cell gets a go-ahead signal in G1, it will usually complete the G1, S, G2, and M phases. If it does not receive it at that point, it may exit the cycle and switch into G0 phase, which is a non-dividing stage. In our body, most cells is in G0 phase. The photo A is describing the process. An example of an internal signal occurs at the M checkpoint. If a cell in mitosis gets a stop signal, that means any of its chromosomes are not attached to spindle fiber. When all chromosomes are attached, then go-ahead signals allow the cell to get into anaphase. By doing this, it ensures daughter cells do not end up with missing or extra chromosomes. From this point, we should make this clear. What are the signaling molecules? Signaling molecules are factors that can affect the cell division. One example is a growth factor. Growth factor is a protein released by certain cells that causes other cells to divide. Moreover, different cell types respond specifically to different growth factors. Let's see another example of growth factor. PDGF is essential when people get injuries. The experiment of figure 12.18 showed that PDGF stimulates fibroblast division to heal the wound. If there's no PDGF, then the cells will not able to divide. However, there are external physical factor on cell division. It is clearly syn and density dependent inhibition. At first, cells in dish surface divides, and then cells form a complete layer and stop to divide. If some cells were removed, the remaining cells divided and filled the gap and completed the layer again. Therefore, scientists concluded that cell density is limited by contact of cells. At last, there is anchorage dependence. To divide, cells must be attached to something. At figure 12.19a, in the beginning, we can see that cells depend on culture to divide. However, there are exceptions of these rules. For example, cancer cells does not matter density-dependent inhibition or anchorage dependence. On figure 12.19b, it shows about how cancer cells divide. Before we go on, I want to make sure about the cancer cells. Cancer cells are cells that divide continually, forming solid tumors or flooding the blood with abnormal cells. Cancer cells and normal cells have certain differences. First, cancer cells stop at the random point at the cell cycle, unlike normal cells. Second, cancer cells can go on dividing indefinitely in culture if there is enough supply of nutrients, but normal cells do not. In 1951, scientists found the HeLa cell. HeLa cells are named HeLa because the original source was a tumor that was removed from the woman called Henrietta Lacks. Through the HeLa cell, scientists found transformation. Transformation causes normal cells to act like cancer cells during the cell division. Now, let's check the tumor. Tumor is a mass of abnormal cells. Then, how the tumor is made. When a cell in a tissue undergoes the first of many steps that convert a normal cell to cancer, then a cell mostly alters proteins on its surface, and the body's immune system destroys itself. However, if the cell evades destruction, it may proliferate and form a tumor. There are two types of tumors, benign tumor and malignant tumor. Benign tumor remains at the original site and does not cause serious problems. This type of tumor can be removed by surgery. However, for the malignant tumor, it can remove to different parts of her body. If an individual has a malignant tumor, it is said to be having a cancer. Recall that malignant tumors can be spread to different parts of our body. The term metastasis refers to the spread of the cancer to the locations distant from the original site by using blood vessels and lymph vessels. In the figure 12.20, it shows the growth and metastasis of a malignant breast tumor. You can check that the tumor is spread by the blood and lymph vessels.